Hello everyone, welcome back to one more video session. In this session we are going to talk about nucleic acids. Though we have studied this concept in our first year PUC, like the types of nucleic acids like DNA, RNA and the structure of the DNA also, and we have learned some terms related to nucleic acids like nucleosides, nucleotides or polynucleotide, all these things. Understanding the basics of all these things like nucleic acids, understanding the structure, basic structure of the DNA or RNA is very important to understand some complex mechanisms like replication, transcription, translation. So that let us go like slowly, will not hurry to finish the syllabus or chapter. Here I have written nucleic acid is the informational molecule. Yes, nucleic acids are of two types like DNA as well as RNA. In some or like in all the living organisms, they have DNA. DNA is itself the genetic material. They are responsible for the expression of certain characters in them. Whereas in certain viruses, they also contain RNA. It could be single stranded RNA or double stranded RNA as their genetic material. One thing is like whatever it is, whether it is a DNA or whether it is RNA, doesn't matter. It contains sequences like adenine, guanine, cytosine, thymine or uracil. It contains sequences which contains information for the synthesis of a specific protein. This specific protein actually has or have a particular function in the body. That's it. So they contain information for the synthesis of a particular protein. So that is the main function of nucleic acid. If you look into the structure of the nucleic acid, it is a long, lengthy structure. It is a polymer of nucleotides. You know what is a nucleotide, correct? Nucleotide mainly contains nitrogenous bases and phosphoric acid, that is phosphate group, and a pentose sugar group, correct? So these are the three things which makes a nucleotide. Many nucleotides join together to form a long polynucleotide chain. If you look into the DNA, two polynucleotide chains are like there. That is actually, it is double stranded. DNA is double stranded, which are linked by hydrogen bonds. Whereas in RNA you don't see them, it is actually a linear single stranded structure, but still it contains the information for a particular protein. So that's what I have written here, that the polymers of nucleotides, it's a like DNA is polymer of nucleotides. DNA is a polynucleotide structure, that's it, it's a chain. Yes, the scientist for the first time, it was before 1900, for the first time a scientist called as Frederick Mischer, he was doing some experiment related to WBC, and accidentally he saw a structure which was situated at the center of the cell, and he could say that okay, so this structure was present within the nucleus, and he called it as nuclein. Yes, he called it as a nuclein. But he actually, the nuclein what he was discovered was nothing but DNA which was present in the WBC or WBC, white blood cells. But he did not know that was the nucleic acid, that was the DNA. He simply called it as nuclein. So please remember it. Frederick Mischer actually discovered this particular nucleic acid, nuclein, and he called it as nuclein. And after that, after certain years, another scientist called as the Altman, he coined nucleic acid term. He called nuclein as nucleic acid. Why he called it as nucleic acid? Simple, if you take a cell, where exactly you see DNA or RNA? Simple, it is within the nucleus. So it is found in the nucleus, so nucleus, nucleic. And whether it is RNA or DNA, it has a phosphoric acid group within them in their structure. So it gives acidity to that particular structure. So it is nucleic acid. DNA or RNA is acidic in nature, found within the nucleus. So it is nucleic acid. That's it. So Altman coined it. Please remember, it could be asked for your NEET. These two scientists. Okay. And next types, you know it, DNA as well as RNA. Okay, so if you take a nucleic acid, let us assume that you are doing some experiment with the DNA. DNA is nothing but a nucleic acid. On hydrolysis, if you break it, you get three, or it yields three components. You get three components. What are they? The three components are, like I have written here, you get two heterocyclic nitrogenous bases. Two different types of nitrogenous bases will be seen, that is purines and pyrimidines. I have written there, purines and pyrimidines. And this is the basic structure of a purine. If you look into the purine, there are two types, adenine, guanine. This is the basic. Okay, adenine guanine with the base structure. This is the base structure, whereas the pyrimidines is this is the base structure of pyrimidine. If you look into the pyrimidine, you see three different types of pyrimidines like cytosine, thymine and uracil. Cytosine, thymine and uracil. Okay, I have not written here adenine guanine in the case of purine. Okay, and next is you will see a type of pentose sugar. Pentose sugar will be there. I have written here. If you are talking about RNA, RNA has a type of pentose sugar. This is the structure of the pentose sugar present in the RNA. What is the difference? This is the pentose sugar of the DNA. Both look similar in their chemical structure, but if you observe it clearly or closely, you see here in the second position you see the hydroxyl group. In the case of, I am talking about RNA's pentose sugar, this is the DNA's pentose sugar. Whereas in the same second position, instead of hydroxyl group, only H is there. This makes the DNA stable. The DNA is very stable because of the absence of oxygen in their second position. Whereas if you look into here, if you look into the RNA structure, OH is there, hydroxyl group is there, which is highly reactive, which is very reactive, highly reactive. As the result, which makes RNA highly unstable. RNA is unstable. That is the reason why RNA cannot be a very good genetic material, whereas the DNA is highly stable because of the presence of or absence of oxygen, and they cannot react with other chemicals, makes it very stable. Obviously, they are now the genetic material. But the first genetic material of the organism during our evolution, like billions of years ago, many organisms, like the initial organisms had RNA as their genetic material, but as the evolution course occurred, it is replaced by the DNA. That's it. So this is the thing, pentose sugar. So what is the pentose sugar of RNA? RNA is actually ribose sugar. RNA ribose sugar, and abbreviation ribose nucleic acid. DNA deoxyribonucleic acid. See, it is based on the type of the pentose sugar they have. In this case, it is deoxy. See, there is no oxygen in the second position, so it is deoxy. That's it. Deoxyribonucleic acid. That's it. It is a ribose sugar. Correct? Okay. Deoxyribonucleic acid. That's it. So this is the thing. And the last one, yes, the nucleic acid, it's because of the presence of phosphoric acid. You know about the phosphate group in DNA, same, it is also present in RNA also. So this is the structure of phosphoric acid, which gives acidity to the nucleic acid. That's it. Okay, so that's what nucleic acid is, bit acidic. So this is actually the basic thing you need to learn. Okay, so let us talk about certain classification of like nucleotides or nucleosides. Okay, so I'll erase it, we'll talk about it. Let us learn about the nomenclature of nucleoside, nucleotide and nucleic acid. You know about the nitrogenous base first. I have written here a nitrogenous base. This is nucleoside, nucleotide, correct, and finally nucleic acid. Nitrogenous bases, there are two types, purine, pyrimidines. If you take DNA or RNA, adenine and guanine, that is the purine, which is common, which is seen in both DNA and RNA. If you take the pyrimidine, in DNA, cytosine is there, thymine is there, but uracil is absent. If you take the RNA's pyrimidine, cytosine is there, uracil is there, but thymine is absent. You know it. This is the basic. You must know it. Whereas if you take the nitrogenous bases, AGCT in DNA, AGCU in RNA. This is the nitrogenous base. Okay, this is the basic thing. Whereas let us go to the next level, that is the nucleoside. This is the level. I am talking about the level. Nucleoside. How nucleoside is formed? When a nitrogenous base combines with a pentose sugar within the nucleotide, that is called as nucleoside. This is the second basic structure. For example, what is the nucleoside? If adenine is present as the nitrogenous base, adenine plus a type of pentose sugar, then we should call it as adenosine. See here, I have not used deoxyadenosine. That means it is RNA. I have used deoxyadenosine. This means it is DNA because deoxyribose sugar. The sugar is deoxy. In this case, sugar is normal ribose having the oxygen. So this is the thing to remember, the basic adenosine. Next level is nucleotide, that is combination of three things. Nucleoside, NB plus PS plus a phosphoric acid, it should be acid now. The nucleotide is, if in the case of RNA, if adenine is there with a pentose sugar with a phosphoric acid, then it is called as adenylic acid. It is also called as AMP. Remember it. Adenosine monophosphate. See, I have written AMP, DAMP. What is the meaning of small d here? Deoxy. That is only present in DNA. See, deoxyadenosine, deoxyadenylic acid. See, if you get like what is deoxyadenylic acid? It is nothing but it is the nucleotide of the DNA. If you get only like adenylic acid, then obviously it is the nucleotide of the RNA. That's it. That is the basic. Okay. So these are all very, very important energy components which provides energy in lot of chemical reaction. For example, ATP, correct? ATP, triphosphate. In this case, it is AMP, the same monophosphate, which also gives energy in chemical reaction. And ultimately, I have written what type of nucleic acid it is, RNA. This is the basic. So you can go through it. Fine. Guanine. I am talking about the second purine, guanine. In the case of RNA, it is called as the guanosine. In the case of the DNA, it is called as deoxyguanosine. That's it. Whereas the nucleotide of the RNA is guanylic acid, that is GMP, very, very important. Whereas the second, the DNA is deoxyguanylic acid, DGMP. That's it. Simple. I have used D, that is deoxy. Here I have not used D, so it is ribose. Okay, so this is in the case of RNA, DNA. Okay, we move to the next one, that is pyrimidine. Very easy. Cytosine, it is present both in DNA as well as RNA. So again, there are two types, cytidine and deoxycytidine. Okay, so cytidylic acid, CMP, deoxycytidylic acid, DCMP. That is RNA, DNA. It is understood. Now, thymine. Thymine is seen only in DNA. The only pyrimidine present in this what you call DNA, but absent in RNA is thymine. If you look into the thymine, oh, I have not written here. It should have been the deoxy, correct? Deoxythymidine. It is also written as deoxythymidylic acid, DTMP, which is seen only in DNA. The last one is the uracil, seen only in RNA. Remember it. So I should not add D here, correct? Only I have to write uridine. The uridine is the nucleoside of the RNA. If uracil is the nitrogenous base, basic. Here you see uridylic acid. It is the nucleotide combination of three things. That is uridylic acid. You call uridylic acid only when the nitrogenous base in the nucleotide is uracil. That's it. The same is applicable to all, and ultimately it is seen in RNA. So this is a basic nomenclature you must know. I have written AMP, DAMP, GMP and all these things. They are very, very important in many, many chemical reactions. In higher classes, you are going to study about AMP, GMP and all these things. But in this year, you are not going to study about it. But I think you will study about the GMP. Okay, guanosine monophosphate, adenosine monophosphate, which is nothing but the nucleotide is adenylic acid because it contains phosphoric acid. That's all about it. In the next session, we will talk about the polynucleotide chain of the DNA and its arrangement.